To our second video assignment of spatial ecology. This week we'll be reviewing lecture and reading material from our second week, specifically on spatial dependence and autocorrelation. First, we'd like to set the scene and redefine a landscape. So as spatial wildlife ecologists, we want to look at the effect of landscape pattern and heterogeneity on the ecological processes that affect wildlife population and distribution. When we ask a pattern process question at a specific scale, the first and most important step is to define a landscape and start a landscape analysis. A small reminder that landscape is not typically defined as a spatially heterogeneous area, but rather it's related to the problem or phenomenon of interest. And just so we don't forget, scale always matters. If our scale is too zoomed in, we won't be able to see the big picture, and if it's too zoomed out, we'll lose detail on our problem of interest.
We also learn about some perspectives in defining a landscape. The two main ones we're going to be concerned with is ecological perspective, which sees the land as an area spatially heterogeneous in at least one factor of interest, as well as wildlife perspective, which sees the land as a heterogeneous distribution of habitat. Some other perspectives we should consider as well as fuel and fire management perspective, geological perspective, and recreational perspective.
There are some challenges, though, to defining a landscape. We have to choose a conceptual model, which can vary from point pattern, linear networks, patch mosaic, gradients, or graph matrix. Additionally, we have to choose thematic content and resolution. These vary in cover types, seral stage, and cover seral stages. We also have to select a spatial scale. It can be either grain, extent, or other types. We also have to account for fragmented features that can happen with different projections and different scales. Finally, we have to define a landscape boundary and context of our problem of interest.
We move on to spatial dependence and autocorrelation. We have to remember Tobler's law, which is everything is related to everything else, but near things are more related than distant things. The example I have here is showing that the species population of the little bluebirds on the left side of the mountain will be more closely related to each other than the species of the pink birds on the right side of the mountain.
Some new terms we learned this week include endogenous, exogenous, spatial dependence, and spatial autocorrelation. Endogenous processes are those that arise directly from the organism or response being measured, like dispersibility, social behavior, or other types. Exogenous variables are processes that arise from outside the organism or response being measured, like environmental gradients. Spatial dependence is the similarity of a variable as a function of spatial location and geographic distance, driven by both endogenous and exogenous processes. Spatial autocorrelation is similar to spatial dependence, but it's only driven by those variables within the organism or response.
So why does spatial dependency matter? Well, samples lose independence of each other and can also lead to type 1 error, which can create hypotheses and biological processes and can alter perceptions of conservation threats.
Type 1 error in this case is assuming that things, specifically variables, are independent of each other when in fact they are not, meaning that one specimen's effects have a significant effect on others within the same study group. Another example of Tobler's law and spatial dependence is the picture here at the right. Here there's a kudu and some black-faced ibis in the Gaborone National Reserve in Botswana. Because these animals are using a shared landscape and a shared resource, and especially because they're in closer proximity to each other, it's likely that they're more related than populations of kudu or ibis that are far apart from each other.